Okay, so we can start the discussion and question session. I'd like to maybe, first see if there's anybody that has a question in the audience to begin with.
So I'm just wondering about the off-target effects that was mentioned during some of the talks and the specificity of the technology. I was thinking about that.
Do I do anything? Hold it for 2 seconds at the bottom.
Yeah, so off-target effects are a concern. They certainly need to be checked for. The idea is that if you're targeting, let's say, a 20 base pair sequence, something very similar may exist elsewhere in the genome, a single base pair difference, for example. You need to know that, and if in fact that's the case, you might not choose that region to put your guide. So that's one type of off-target that you need to look for. Surprisingly enough, that seems to be more of a problem in some of the iPS cells. And really in a lot of the work in mice, in vivo, they can't find those at all. So it's a little bit controversial right now why that differential should be, but it's something to consider in your designs for sure. The other kind of off-target I was talking about is just plain a non-therapeutic cell. In other words, you're trying to deal with a blindness disease and you don't need it expressed in the muscle around the eye. And that's just sort of a general principle of any therapeutic. You want it focused where you would like it to be.
But presumably there could be, I guess, non-specific cuts made and you would be unaware of them.
Although we're going to be developing assays so you're not unaware, you can, so let's say you choose 3 or 4 guides that cut very efficiently, and then you characterize each one of them for their off-target cutting. And you then choose the one that has the minimal off-target and the maximal on-target. So it's part of your design strategy to minimize that.
Maybe before we go to the next question, a follow-up point on that, and that I think we can address also at the final panel on regulatory issues. So we all know that drugs, the ones that we're taking today, the ones that are approved, many of those have, I'll say, it's not really off-target, but many adverse events that will come together with the drug. So how do you think will be the regulatory landscape? Is it going to be more severe, or is it going to be more for CRISPR and just say we're not allowing any off-target effect, whereas today we have drugs that do have adverse events. So
I think when something's new, one is very cautious. These first clinical trials are phase 1, primarily safety. Safety will be concerned. The major worry about off-target is that you accidentally induce an oncogene and create a tumor. Nobody has seen that yet in cells, in mice, and so that's why these clinical trials are getting permission to move forward. But of course, the first thing is safety, and they will be held to a very high standard.
Yeah.
I think another aspect of that is where some of the initial attempts to use CRISPR in human health have targeted things that are otherwise lethal in the first couple of years of life, then you're not so worried about off-target or non-specific effects in those cases.
yeah, question.
In a related issue on the biosafety, my question is that how stable is it within the same generation? But more importantly, when this person reproduces, what happened to the CRISPR-altered gene, the penetration or transmission rate or stability? Do we know any effect on that?
All the current work for therapeutics is called somatic cell. So the target is a cell of your body exclusive of your germline. So you protect the germline, you don't go there. And so there would be absolutely no passing on to the next generation. If I have sickle cell anemia, I still can pass it on to my children. It might be cured in my blood, but it's not cured in my germline. I can pass it on to my children. So these are non-germline therapies that we're talking about. They're somatic cell therapies.
But your genome, after you edit the gene, is still one of the chromosomes being...
But unless my sperm or eggs are changed, then I don't pass it to the next generation. And so they're not being changed. My eye, my blood, my muscle, those are being changed, but those aren't my sperm and eggs. So no, it only affects me, no future generations.
And the stability of the edited gene, the example that you showed, are you just lucky to have 7 years or whatever in the system, health system or human system that you're using that's stable? But can you generalize it in other genes once edited it that is actually made stable? And referring back to that gentleman mentioned that about the specificity of the edited gene.
So if I understand what you're asking, gene therapy in general has kind of solved the problem of turning off of the genes. So for example, in a dog, they have a 12-year extended treatment where the dog was treated and the therapy worked for the full life of the dog until it was euthanized when it was 12 years old. So I do think it's much more stable than in the past. And that would be ideal. Gene therapy is kind of ideal. Gene therapy is one and done. We treat you and we never see you again. And much like replacing your heart or something like that. And so that is the ideal. It's not always achieved. Some of these ideas, there will have to be follow-up treatments, but that is the ideal.
One follow-up to that would be, you can imagine targeting certain tissues that have turnover, and there you might need repeated treatments. But if you're treating something that's terminally differentiated, like the retina, then you either are going to lose the only way to lose the tissue that has been changed is for it to be destroyed, and there's no way to replace that. So. Yeah, it should be one and done unless you're looking at something that affects, for example, the gut lining or maybe blood cells, something like that.
Maybe one question, and you had a comment, but one question for Vincent, Eric, more on the synthetic biology side and the commercial and application opportunities. So we have you here on the panel for health. But I know both of you are somehow involved in all the sectors, either with synthetic biology or CRISPR. So I would like to hear from you what you think are the low-hanging fruits. Where do you think we can see shorter-term benefit in general, social benefit and economic benefit from these genomic engineering technologies? So maybe you can go first, Vincent.
Sure, I can take this one. So yeah, in reality, I do a little bit of health work, but most of my work is somewhere else. The two examples that I showed for these companies that are starting up, they're really looking at food, they're looking at drug production, they're looking at small molecule chemistries. A lot of these things don't require FDA approval, they don't go through long clinical trial periods. So those are, I think, the early places where it's going to make a difference. The issue, and I alluded to it briefly, is if you're producing something, if you're producing a food elements of some sort, you're selling that for not much money, so your cost of R&D and development has to be low. And this is where the issue and where synthetic biology comes in right now is it's difficult to imagine investing hundreds of millions of dollars for selling something that has not much value in a sense. So by applying this kind of technology, you can really speed things up, accelerate things, and the predictability and the speed. So you know that whatever you do, the outcome is going to be what you intended, and it's going to be fast. And all of a sudden you can kind of start going down on the value of the molecules you're selling. And this is, when we talk about biofuels, this is part of the problem. Biofuels, large volume, low value. So it's a tough target to go for.
I think in the slide that I presented, the applications, it really, I think, does follow that in terms of even near-term applications. In basic research, that's having the greatest impact right now. All of the academic researchers that are using the technology, let me add briefly too that all of the IP holders are not looking for any sort of reach through into the commercial side on the academics. So academics are free use of technology. But when it gets to a commercial application, I think the near-term things that are happening are healthcare. you're actually seeing most of the investment in that space because of the return, and so that's moving at the fastest pace. And then I think I mentioned industrial biotechnology, and there it's because there's no regulatory environment that's really holding that back. So the commercial application at least is moving forward. And I think we'll see that begin to change in the agricultural space. There are already filings on that side, and then moving sort of up the chain. There are animals in the agricultural space, dairy cattle that don't have horns and things like that, but it takes actually years to breed up a herd of that kind of cattle. So I think it will take a little bit longer before we actually see an impact from those areas. But all of those areas are being pursued at an incredible pace right now.
Any questions from the audience? Yeah, and maybe for the questions, if you can introduce yourself first, so that we also know who you are.
So, good morning. My name's Tom Digby. I've been one of the people for 20 years waiting for gene therapy to happen, and it hasn't happened yet. It's the technical approach you described, Elizabeth, is fascinating with CRISPR. But the delivery tool, the AAV vector that's used to deliver these in vivo therapies, still seems to be not perfected yet. So I'm wondering if you could comment. Ex vivo therapies, I totally agree, are going to lead the way, but are in vivo therapies really coming along?
So they're here. So almost every child with LCA deficiency now in the United States has been treated with AAV gene therapy under a research clinical trial. And that is now hundreds of individuals. And if you watch the videos, they do a video of the child manipulating through a complex laboratory jungle of obstacles, and then they do it again a year later after treatment, and the child has no difficulty. Now, so it's here for a small set of diseases. It's here. AAV has been an incredible breakthrough. I don't think we're finished developing it yet. I think it can be further improved. There's lots of research to do, but it's good enough to already be in the clinic and doing good for children. I work at a children's hospital, so I'm mainly focused on that, but there are adults being treated as well.
I'm Robin Coop from the Genome Sciences Center. I had a bit of a nebulous question for Vincent. What is the sort of cost of doing synthetic biology experiments? Like, broadly speaking, we have a sense of genomics experiments cost, a couple $1,000 a lane, and we can do whole genomes in that range. I mean, I know it's a big, complex question to talk about analyzing, complete patients and so forth, but we have a sense of those costs. And so we go do genomics experiments and we get hundreds of thousands, a few $100,000 or something. If I want to do things with bacteria, am I looking at having to get millions of dollars, or can I do it for sort of Canadian scale research funding, or where where is that at?
So I'm going to answer that in two ways. So it depends on scale, for sure. So in my lab, we hardly. I don't think I have ligase in my freezer anymore. We don't use that stuff. I don't even clone DNA anymore. We just call these companies, we get everything by mail. I mean, they're still selling at 8, 9, 10 cents a base, but if you start calculating your cost of reagents, your time for your students, your failures over and over again, we pick up the phone, we have 100 clones in a couple of weeks in the mail. So to me, I think that insert CIHR level grants, you can do that, and it speeds things up quite a bit. So we regularly sample 10, 20, 30 kb of DNA that way in yeast. So I think that's totally achievable, and I see a lot of my colleagues are starting to go that way. If you're if you're now starting to think about a bigger scale, some of these companies will give you a discount, but I mentioned the GP Wright program, the grand challenge that they're having. They're modeling this after the original Human Genome Project, where when that started, the cost of sequencing was not there at all. And one of the big pushes that the NIH had was to go for that $1,000 genome sequence project. So they're establishing the same grand challenges now to reduce the cost of gene synthesis and synthetic DNA to fractions of a pennies per base. So that's part of the challenge, is to really lower the cost. And there's technologies coming online. There was 2 companies there in New York last week where they're looking at using enzymes to synthesize oligos as opposed to your traditional chemistry, which is much faster and much cheaper. So I think some of that technology is going to come online in years to come, and it's going to get dirt cheap.
Yeah, maybe one comment and a question following up on that. So we're talking before we started that if we think about the whole Human Genome Project that used Sanger sequencing and costed $3 billion, which is incredible when we think today that we can sequence a whole genome for $1,000. So what's your vision? How long would it take because as I think today, and what we're discussing here is that basically we are at the Sanger sequencing stage for synthetic biology. We're not yet there with the right technology to really deliver as we are able to deliver in producing or writing genomes today. So how long do you think will take? and what will be the next steps now to really get there? What do we need to do? And back to your comment also, do we need to invest millions and millions, and can we? Is there a light at the end of the tunnel for that technology?
So I'm going to try to read the tea leaves here. Again, at this GP Wright meeting last week, they were trying to identify what the costs are now. So in a sense, if you actually look at the cost of DNA synthesis today, we could synthesize a human genome for less than $3 billion. So the challenge is actually not as big as it was for the Human Genome Sequencing Project. We could just brute force it and get it done. I don't think that's a very elegant way to do things. How fast is the cost going to go down? So there's 2 limitations. One of them is oligosynthesis, and I think that's going to go down pretty quickly. There's a lot of technology that are online right now to do this. So one of them is miniaturization to get higher scale, but the other one is brand new technology coming on, right? The other issue is you're not assembling 1 or 2 kbs anymore. You're assembling 500 megabase pairs at a time. If you hope to achieve the synthesis of a large eukaryotic chromosome. So that's a challenge. You're talking about a 500 kb plasmid that you're moving around. It's not an easy thing to do, so there's challenges on that side. The biggest that's been done is the Venter Institute, about 500 kb genome in the yeast cell. So putting these small pieces together and assembling is the next technology challenge. And on the synthesis, of course, and if you've done any of this work, the minute you start getting into a lot of repeats, synthesizing that from scratch is going to be a problem, technology problem. You try to eliminate those as much as you can. I don't know if that's going to be possible for higher eukaryotes and complex genomes. But the cost, I don't know. I'm hoping fractions of pennies for sure, but it's that exponential curve. I don't know where it's going to go.
Yeah.
Okay. So it's a
follow-up question. So my name is Renee Warren. I work at the Genome Sciences Center. And I've been there for almost 15 years. But I've been involved in biotechnology for almost 2 decades now. So I guess related to this point, so you mentioned, Vincent, that we're very good at reading genomes. So we can synthesize genomes. So I've been involved in that, some of that work as well. One of the things that I guess realized over the last 2 decades is we're not really good at finding out what various components of the genomes do. And so that's, I guess, problematic for synthetic biology, because I'm just thinking about some of the slides you showed on the Craig Venter work, where they essentially synthesize a minimal genome. And so they had to actually figure out what every single gene did, and they couldn't figure out what most of them did to put together that genome. And that's great. I mean, these are great achievements. But I think we'll have to invest a lot more in trying to figure out what the components do.
So maybe I can answer that. The pharmaceutical industry didn't start knowing everything about the human body before they started messing with it. So, right? And
it's not so much an
it's-
issue for CRISPR work.
And it's that's why they have clinical trials and most of them fail, right? Because you realize the molecule you did is not targeting the right thing and it's not having the right effect. So the model for synthetic biology is the same. You're going to have a lot of failures. And I mentioned the genome design. So So really what you do with the software is you give it rules to the chromosome you want to build. And initially, and this is what they mentioned in this Saccharomyces genome, is they actually had very stringent rules. They were hoping for success, so they only did a few little tweaks on the genome to realize that genome is a lot more plastic than they think it is. And the other thing, too, to talk about is, in the human genome, only a small part is coding sequence. If you're going to go after the non-coding sequences, that's a completely different problem. But again, you can start attacking it piece by piece. And I think that's a term that's very often used in cellular biology is you learn by trying to build these things, to start putting these things out, these things. That's what Leonard did for his minimal genome, and see what the effect is on a phenotype. So yeah, we don't know everything, but it shouldn't prevent us from going forward and doing some of this work.
I think what you're really arguing for is more basic research, which I think we can all get behind.
There's also the cell-free systems, so depending what you're doing. So if you're working on some biotechnology project where you don't need all the excess baggage of a cell, you can do stuff in cell-free systems, and those have been developing a lot in the past few years. So I think there's going to be a lot of opportunity in that area.
Any other questions? And well, for those of you who are very shy to press and ask your question, you can always ask your question via Twitter. I know we're following you and you're following us, so tweet your question and we'll be able to ask your question. If you're shy to come up with a question.
I have a burning hand. So I've never been in.
Someone else has it on.
Okay, so I've never been involved on the, business side of things. But what I'm wondering is, if I want to do CRISPR work right now, and I guess I can order the tools from Harvard, MIT, from what I understand, it would be quite easy to get. But if I do come up with a really cool technology, how do I go about and get this to the next step? Not just publishing on it, but
yeah. Well, before you publish,
ABC of creating a company
Yeah.
in 5 seconds.
Sure, 5 seconds. Before you publish, patent, and then publish, and then people will either find you or you can seek ventures you can go with. There's lots of translational assistance. I'm not familiar with BC's system. In the US, a lot of the universities have systems to help people engage in that. But essentially, if you're going to commercialize and you're using somebody else's patents, the CRISPR patents, you would have to go to one of the people that I outlined there, for example. But you may have an add-on to that. That's where the value is for your company.
I see. But none of these IPs prevent us right now, by the sound of it.
Not at all. Not at all. And I mean, a little plug for ERS. Our goal is to get the technology out and used by as many people as possible. So we're making non-exclusive licenses, I hope, reasonably available.
And Eric, maybe a comment. Your company is based in the U.S., but somehow started here. Tell us a little bit about that.
Our company is based in Ireland.
Okay. Oh, yeah, true. You're based in the
I'm in the
U.S.,
US.
and the company somehow started here.
Yeah.
Or?
One of the owners is from the Vancouver area. One of the founders. The other two, Emmanuel Charpentier is one of the co-discoverers of the technology. She lives in Germany now, and then our other founder is living in Switzerland, running a company based in Boston. We're kind of all over the place.
And you live in California, so it's quite an international business. Yeah, Rachel.
So I just wonder if any of the panelists could comment on the potential for CRISPR to be used in germline applications. I mean, I hate to bring it up, but we're talking about things that are in the future, and it's being done in some countries.
So we must discuss it in relationship to the animals. For sure, those are germline changes, and that needs continued conversation. As far as I know, and I really only know North America and England and France and stuff, it is completely banned and absolutely unfundable. You would get nowhere with it. But that doesn't mean it shouldn't be discussed. And I think one of the things I want to underline is people say CRISPR is easy. I think that gives non-scientists the impression that, you can do it in your bathroom or your kitchen or something. It's easy compared to talons and some of these meganucleotides and ASEs and stuff. It's not that easy. So you aren't talking about people doing this in their kitchens. But right now, absolutely not any human application for germline at all, either funded or even ever considered for a clinical trial. But in animals, absolutely, that's exactly what we are doing, germline changes in animals.
But I think what we do have to consider is it's possible now. So it's an ethical discussion at this point. And you're right, none of the scientists, I don't think, want to proceed with that right now. But it's certainly a viable approach to germline editing because it works in animals. So we have to consider that.
I do think consideration is important, but just to say not even a monkey has been made yet. So to say it's doable, if you're actually the one doing it, it's hard. And so not even a monkey, mice, pigs, rabbits, that's why I showed those. So we're not even in primates yet. But they're working on it, of course. A monkey model of a rare genetic disease would be an incredible tool for curing that disease. So it needs discussion. But just to say, it's not easy, and it's not around, it's not like I could do it today. I couldn't, even if I wanted to, which I don't.
Maybe a follow-up question. Yeah.
It's a bit relevant. Anthony Chan, I'm a clinician working in human reproduction. You can see the reason why I asked a question about transmission of the edited gene to the offspring. I understand your point. We're not working on the germline, but you still alter the somatic gene in a chromosome. So that person is cured. Now he or she is in better health. She subsequently going to conceive. What happened to that gene that get edited during fertilization, meiosis? Is it still stable? Or you have other effects? What about the genetics, masking and unmasking the expression of the gene?
So we really need to get the concept of somatic versus germline. So if those children who are being treated for blindness, that change in their genome dies with them. They can have a family and it will be just the way it was going to be as if they were never treated. And in fact, because their sperm and egg was not modified, the only thing that was modified was their retina. And so in that sense, they are going to pass on their genetic disease to their children because their sperm and eggs were not changed. So if what you mean is permanent in the next generation, absolutely not. Permanent in the eye, perhaps, we hope, for the life of that child. But that change will die with that child. We could talk off. I think still that somatic germline difference must be clearly understood.
So you're working with somatic cells?
Only somatic cells. Only somatic cells.
Mohammed?
Hi, it's Mohammed from Genome BC. So I just want to follow up with that. So say in somebody who conceives, doesn't normally like antibodies and other things pass from the mother to the child in utero? And is it possible that something like this could also pass through? Like if it's treated, how is it treated? Is it treated very locally, or is it treated in the?
So there are different strategies going on right now. Most of what's going on is very local because that's what gets permission. That's, for example, why the eye is leading the way. It's because it's considered a closed, very safe place to put anything. But it's kind of like asking me whether we don't give mothers drugs that would pass through and damage their children. In the same way, it's absolutely against the idea of gene therapy that anything that you did to the mother would pass through to the child. And so the answer is no. Have they tested it? Have there been lots of cases of pregnant moms who were treated? I don't know the answer to that. But certainly in the animal models, that doesn't happen, first of all. And if it were to happen, it would be a complete ban for that, would not move forward, just like thalidomide shouldn't have moved forward. Yeah.
Maybe to move a little bit from the somatic germline debate, and feel free to follow up during the break. We'll have plenty of time during the day to have one-to-one follow-ups. So some of you talk about, I think, Vincent, you talk about potential applications in malaria, infectious diseases. There's also this concept that we're going to talk about in the next panel about the gene drives and the potential role in infectious diseases. So there we're thinking about a global and global application that could have a public health impact. But at the same time, you were talking, Len, about your HLA example, where you were thinking about almost that you might need to develop a CRISPR for every individual. It's so diverse. So I guess my question here on the synthetic biology side and the CRISPR side is how global can we be, or do we need to develop very specific technologies for every one of the problems, or are they like the gene drive, that can kind of overcome and really, and we'll talk more about that, but how specific do we need to get in terms of synthetic biology and in terms of gene editing, or can we think about more global solutions?
You're on. I don't know that there's an answer to that. I think for some things, and probably the reason we've seen successes so far is that people have gone after diseases where it's in a relatively stable and polymorphism-free area of the genome. But I would think as we move, one component of CRISPR applications in human health is probably going to be having your genome sequenced ahead of time to ensure that it's going to target properly. And for some things, especially in autoimmune diseases where there's many different potential targets, there's probably going to have to be a lot of investigation in the individual to figure out what the mechanism is first before you design the CRISPR. So it's not going to be an off-the-shelf thing that you inject into someone. For some things it is, but for others it's going to be it's going to involve a whole medical and lab bench team to figure out what the strategy is going to be.
So maybe I can add a little bit to that. I don't think there is an answer, by the way, but I'll try anyways. I think what's central is the technology and the tools, and the individual applications are going to need tweaking. But a good example for, let's say, synthetic biology is repurposing codons, or sometimes they call it XNA or something that's just for biocontainment. So we realize, and you mentioned industrial biotechnology, it's one of the really ones to take off because you're growing this in a fermenter in a very controlled environment. But as the field progresses, it's going to go and be more outside of our control. So a lot of people are thinking about biosecurity and biosafety. So an example I think that was brought up at one point is you mentioned is adding microbes to the roots of plants so that'll fix nitrogen and help them. So if this is a genetically engineered microbe, you could ostensibly repurpose its codon and or give it a non-natural amino acid so it's genetically contained, and its genetics can't be passed on. So the technology to do that would be similar across what you're trying to do. There's been other examples for vaccine, let's say, but you're going to have to engineer that microbe specifically every time you want to do something. The idea here is to accelerate and speed things up with the technology, so you're not spending $100 million every time you want to do it.
One Twitter question.
Twitter question. Yeah. So this comes from somebody in the audience. Are we starting with the retina because of less immune kickback from AAV therapy?
I missed some of that.
Sorry, I got to read. Are we starting with the retina because of less immune kickback from AAV therapy?
Absolutely.
Absolutely. First of all, one of the reasons AAV has been so successful is it naturally evades the immune system. So if you don't mess with it too much, you have that as an advantage. Also, the virus doesn't replicate in the human body. The modified recombinant version doesn't replicate, so it's not producing viral proteins continuously. It's a bit like being vaccinated. You get the proteins once and you don't get it again. The therapeutic protein does go on, and it has to be produced continuously. That's the whole point. You're trying to deliver that. And the eye is a particularly immune tolerant site. And so it is leading the way for that reason. The brain would be similarly immune tolerant. None of them are perfect. This is absolutely a concern. But for a lot of these diseases, you may already, well, the one I work on, let me use my example, aniridia, the individuals have the PAX6 protein. They just don't have enough of it. And so we won't have any immune response to PAX6 at all. All we're doing is augmentating the natural protein. So that's kind of a low-hanging fruit example. And many people are challenging themselves in that way. Deliver a therapeutic that is not novel to the body.
Chen.
So this is Chen from Genome BC. I'm just curious if you could comment on the non-viral delivery of CRISPR-Cas9 system, such as lipid nanoparticles.
So really the answer is no. I'm no expert on the non-viral delivery. I certainly saw talks at this most recent meeting I went to. They were certainly being discussed. Nanoparticles and RNA protein particles were being used. They have significant delivery challenges of their own. My take would be it isn't a one method fits all diseases. I think it's very much going to be tailored to individual diseases. So all those sort of three would be leading the way right now, those three approaches. But of the three, the viral is the dominant one at this time.
I would just say viral is dominant for the in vivo setting. I think ex vivo, that's not necessarily the case, and it's very cell type dependent.
Vasily Triandafilidis from Chemical Biological Engineering. So I have a question for Dr. Rhodes. Imagine I want to commercialize the technology with CRISPR. I license out the technology from the people you said. What are the major hurdles in terms of the FDA approval? What do you think needs to be changed, maybe in future, for more streamlined approval? And what do you think is leading, which countries are leading in terms of regulatory approval in the world?
That's a very broad question, but I'll try and focus a bit. I think as far as countries that are leading, I think the US and Europe are on the case, I guess I would say. I'm not sure that you can actually say that anybody is leading. I know I'm involved in a clinical trial that will take CRISPR ex vivo into the body this year in the US. That's great. I've heard similar things in Europe. In China, they've already treated humans with an ex vivo therapy. So I don't know the regulatory status of it there, but they are ahead in terms of actually treating. So I think, and everybody kind of takes their lead from those organizations. I'm not sure if Canada is any further ahead, but I would say it's probably right, essentially at the same level that the other organizations are. The people that are leading the charge are the people that have their therapies ready to go. Most of these are investigators in research institutions and not necessarily the companies. So you will find individual researchers getting into the clinic faster than these companies because the companies are pursuing a commercial angle. They can't have an FDA setback of their investors all flee. So right now, most of this is being done on a more of an individualized basis at research institutions, and they are therefore sort of setting the guidelines that the rest of the institutions and companies will have to follow. But the FDA, the MEA, they are all looking at each of the applications individually, I would say. So ex vivo sort of leads the charge. There will be in vivo trials very soon. I can't tell you whether it will be Europe or the US, although Europe tends to lead in that area. So I don't think there's one answer to what you're asking, but there are already many programs in play going through the regulatory system, and you basically would look at those for guidance on how to approach yours. If it's an ex vivo, if it's an in vivo therapy, if it's prolonged expression, if it's a one and done genome edit, things like that.
We have 2 questions there.
If I can make this one. Hey. Hi, I'm Shweta from CBRD, and my question is a bit broad, and I was just wondering, has there been any work done on using gene editing technologies to enhance your immune response to infectious diseases? There's some work coming out of Dr. Janelle Ayer's lab in Stanford saying that your immune system actually responds in a certain way to infectious diseases. I was just wondering if there was any work that you were aware of.
Everybody's looking at me, assuming that I have an answer. I don't have an answer. I am not aware of any group that's doing that, but I think it's partly a case of us really not understanding the immune system well enough to make those decisions. Unless there's, there's cases where someone has a rare immune disease where actually it has involved CRISPR, but where it's been treated at a molecular level in various ways. But those are not quite what you're asking. I'm not aware of anything that kind of fits what you're asking about, but I could be just naive there.
The one thing I can say, there's a lot of interest in CAR T therapies, and while that's not infectious disease necessarily, it is a bit of an approach towards monitoring the immune system, maybe making it less tolerant, more tolerant to certain antigens. So I think there's some parallel work going on. And then there's the HIV work that has been done. That was Sangamo has got a program to actually modify some of the T cells within HIV patients, make them resistant to further infection. So there is that work.
So I was actually talking to Rob Oatley yesterday, and he was talking about his CAR T work, and one of the things that he mentioned, which maybe speaks a little bit to what both of you are talking about, is their ability to deliver bigger payloads. Once you start making much bigger modifications and having and touching more different aspects of transcription regulation, whatever, you need to put more DNA or affect more things in the cell. And at this moment, what Rob tells me is that that's a limitation. And that's one of the things that we're open to overcome.
And we have that, I think this is our last question. Yeah.
Well, I'm Michelle. Anita Howe from Center for Excellence in HIV and AIDS. I have 2 questions. The first question is that from the side effects perspective, can one repair the repair system? Meaning, like if you have some untoward biological surprises after the gene therapy, and can you repair it? The second question is that being brought up in primarily pharmaceutical discovery and development, and one of the biggest things that when we develop drugs is the PK/PD. So can the system that you describe, is it scalable? Can you adjust the different magnitude of effects?
Okay, thank you. Who wants to start? Scalability, and can you repair the repair system?
So deal with scalability first. That is a big challenge. And there, I agree with what was said before. A lot of these clinical trials are being led by academic institutions. But with success in the treatment of more and more patients, you have to shift to a more production-like, larger scale. And I can't speak to that. I'm only aware that that's an issue, and that each therapy has to deal with it for their own, to move forward. As far as, what was the first question? So scalability and repair the repair. I haven't heard about that. There is a huge amount of discussion about off switches. So if you put a gene therapy in right now, one of the positive promises of gene therapy is that it would be like, would be a treatment that is one and done and lasts for life. But the FDA would like off switches in case there is something bad that happens 10 years later and you would like to turn it off. Generally speaking, the field has not solved that problem yet, and these do not have off switches. They're on for life. I think that's partly why it was started with the eye. At least in my field, always the most blind, the worst eye was treated with the concept that if in fact you had already lost vision and something terrible happened, you could then surgically remove it. Let's say it became cancerous. That never happened, so it hasn't happened in the field, but I think that's partly why a site like the eye led the way for the nervous system. But the off problem is a big problem in the field, and it has not been addressed. It is not currently holding us back, but I can see that it would be wonderful to have if we had it.
The challenge with an off switch is it requires additional material to get delivered, and now we're back into the realm of problems with delivery. So there have been off switches. They haven't been needed, to my knowledge, although in the childhood leukemia trial, you would say it would have been nice to have one, and they didn't. But that was also a modification that occurred at another site. It was an off-target modification, so you couldn't have predicted that. So it is a problem. There are ways to address it, but the ways of addressing them sometimes prevent the therapy from being effective. So there isn't a great answer yet.
Perfect. So I think with that, I would like to thank the speakers for keeping time and being engaged with the discussion. So join me thanking the speakers.